Okay, this is round 2 here at ThinkTech today. This time it's ThinkTech Talks in Hawaii, the state of science, okay, with Matt Koenig, MD, who does research at Queens Hospital and at JABSOM. And if you don't know what JABSOM is, I'm going to tell you for the 150th time. It's the John A. Burns School of Medicine, okay? Welcome to the show, Matt.
Thanks so much for having me, Jay.
Thanks for coming down. We're going to talk today about your MRI research, which you do at Queens Hospital and I suppose at JABSOM too, but I recall, and I want to just confirm my recollection is right, that in the basement of Queens Hospital, down where nobody ever goes, maybe 2 levels below the lowest level you ever went to, there's this MRI research facility. And it was where I met Linda Chang and Thomas Ernst, who live down there and do MRI research. And you gotta watch out for your keys. If you don't hold your keys carefully, they fly across the room and smack into the MRI machine.
It's actually a very lovely facility on the ground floor of the hospital, directly across from the parking garage. But
Oh, okay. They must have moved it.
not in the deepest bowels of the hospital.
Okay. So this is a facility with a very expensive MRI machine. You want to talk about the MRI machine there? I guess this is where you're working, yeah?
That's right.
Okay.
So a little bit about the MRI research facility. It's run by Linda Chang, who's a neurologist, and Thomas Ernst, who's an MRI physicist, and they are my mentors.
They both came from Brookhaven together.
That's right.
It was research. It was a romantic research relationship.
Right. So I've been in Hawaii for about 5.5 years now, and the research center was very well established by Drs. Chang and Ernst by the time I arrived, so I can take absolutely no credit for that lovely facility, but I've definitely benefited from it in terms of having excellent research mentorship. And so but the MRI research facility is actually jointly run by Queens Medical Center and JABSOM. And Dr. Chang and Dr. Ernst and the other PhD researchers and MD researchers in the organization have appointments at JABSOM, which is the John A. Burns School of Medicine, as well as working in the facilities at Queens Medical Center. Mm-hmm. And I don't know all the financial aspects of it, but I do know that the research facility was partially funded by Queens, and the space was donated by Queens. However, the research group there is primarily affiliated with UH JABSOM. And so I actually have appointments in both the medical school and the position at Queens Medical Center.
So you're a research doctor. You don't see patients.
I do see patients.
You do see patients.
So primarily I see patients.
Oh, is that right? You're a clinical doctor first.
Yeah, I'm what I would call a clinician scientist.
So how much of your time do you devote to one and how much to the other?
Well, that's, we can spend some time talking about the state of research in Hawaii and how you can get researchers to move here from the mainland. And the answer to that question is really the story of why I chose to move to Queens Medical Center from Johns Hopkins, where I was on the faculty. And it's in part because of the support that I received from the administration at Queens, where they allow me to do my regular clinical responsibilities, and I'm salaried based on my patient care duties. However, they also contractually protect some of my off-service time when I'm not treating patients, so that time can be spent doing research.
What do you mean protect?
Protect meaning that time cannot be taken away from me for clinical responsibilities that are unscheduled. So I have scheduled on-service time. For example, I'll be on service in the intensive care unit starting tomorrow, and for the next week, I'll be primarily taking care of patients. However, beginning next Tuesday, I'll be off service for a week, and that time is completely protected for me to do research.
Well, that's great. This is what you wanted.
Right. That's kind of the holy grail in a lot of ways, is that you can derive your salary from patient care. However, the institution protects your time, so it's not in jeopardy. So you can be, in jeopardy meaning your research time can be taken away from you to do patient care responsibilities without notice.
So what about what about the kind of clinical work you're doing? I mean, what is it? I mean, are you board certified in one of those clinical specialties?
Right, so I'm a neurologist by primary training, so I'm a board certified neurologist, and then I have additional subspecialty training in vascular neurology, which is stroke. So I'm a stroke specialist. And I also have board certification in neurocritical care. And basically what neurocritical care means is that I'm an intensive care physician who takes care of patients who are critically ill because of neurological and neurosurgical diseases. So diseases like severe strokes, very bad seizures, or bleeding in the brain, or aneurysms, or neurosurgical conditions, or severe injuries of the spinal cord. So any condition that relates to the brain and nervous system that can result in critical illness, that's what I specialize in.
So is it just an accident that all of these things require sophisticated MRI testing?
Well, I wouldn't say that all of those conditions require MRI, but MRI is certainly a very useful structural tool to look at injuries to the brain, various types of injuries to the brain, and is a very helpful diagnostic tool for neurological conditions.
So you're a regular doctor too, so I can call you up.
Yeah, so I'm a regular doctor, and I think the research project that I do, I do several research projects, but the research project that we're going to talk about today, based on functional MRI, is really based on my clinical experience with patients. And I think the best translational research really comes from the bedside, meaning taking care of difficult problems in patients at the bedside and not having the answers to those problems, and having those problems, carrying those problems with you as you see patients, and then designing research projects to try and answer some of those difficulties for patients.
So when you say translational, that's an important word. When you say translational, you mean translating from the clinical bedside into the research facility.
Correct. So translation meaning bedside to bench, and bench back to bedside.
Ah. Well, that's
or
really...
in my case, I guess there's no bench, there's more of a powerful magnet.
Right, right. It is the one with the keys, right?
Right.
Where the keys, fly across the room and don't get in the way?
That's right, yeah. And don't take your credit cards in there either because they get erased.
Now that MRI, that's quite an MRI. I recall when I talked to Linda Chang and Thomas Ernst, must be 10 years ago, that cost millions of dollars. They designed it, or they selected it from the manufacturer. It took a long time to get here. It took a long time to install, but it is a very important piece of gear to do MRI research because it's a super MRI.
That's right. So it's a 3 Tesla magnet. The standard clinical magnets are 1.5 Tesla, so it's double the magnetic power, I guess you would say. Thomas would correct me, I'm sure. But double the magnetic force of a typical clinical magnet.
So this is very important for Hawaii science, for medical science anyway. And it just to take a moment and say, you were a speaker in our program last Thursday, January 22nd at Laniakea, which we called Hawaii, the State of Science. You spoke about your research and showed us, as we knew, but we want the public to know, that there really is scientific research going on here that is world-class scientific research. In fact, I would say that it is a very important segment of the pie of all the research that goes on in Hawaii. People have to know that, they have to understand that. Anyway, so let's talk about, exactly, well, let me preface by saying, I remember that Linda Chang, when I interviewed her, was doing the brain. Mm-hmm. She was doing sexually transmitted diseases, I don't remember which ones, and what effect they have on the brain, and how you could see what was going on using the MRI. And she had a lot of graphics to show the way the brain deteriorated in the face of those diseases. Yeah. Is your stuff like that, or is it different?
Well, Dr. Chang, my mentor, does a lot of very broad research using functional MRI. Functional MRI is really a tool that you can use to study a lot of different disease processes and also normal physiologic processes of the brain. And so her major areas of research interest are HIV, the effects of HIV on the brain, and how the brain atrophies in relation to HIV, and changes in the function of the brain related to HIV. She also has a major avenue of interest in studying the effects of drugs of abuse, specifically methamphetamine.
I remember now, yeah.
Methamphetamine on the brain and how brain function changes in chronic users of methamphetamine. And she also studies normal aging, looks at the structural and functional changes of the brain in normal human development in pediatrics. So she has a lot of different research projects going on. and mine is just one of those projects.
Which one is yours?
So I study cardiac arrest survivors, so people who have had cardiac arrest. And what I mean by that is an arrhythmia of the heart, an abnormal heart rhythm, or stoppage of contraction of the heart that occurs suddenly. And there are various causes, potential causes for that. But suffice it to say that cardiac arrest is a period of time where a person's heart stops, and the person is not breathing, and they have no pulse. And during that period of time, there's no blood flow and no oxygen going to the brain. And so in a lot of ways, it's like the world's largest stroke. It's a complete blockage of blood flow to the brain for that period of time. And as I'm sure you're aware, CPR has been a tried and true technique that the public is becoming more and more educated on how to do compressions of the chest and assisted breathing in order to support blood flow to the brain and other vital organs during the period of time when the heart is not beating. In addition, AEDs, or automatic electrical defibrillators, are widespread now in the public. You see them in the mall, airport, lifeguard stations, and various public places. And so what we're seeing is that more and more bystanders or lay public are being trained in doing CPR, and there's greater availability of defibrillators. And so the percentage of people who are able to be resuscitated in the field, meaning the heart is able to be supported by chest compressions, and then shocks are delivered, which restore the heart to the normal rhythm, and then the heart begins pumping again and blood flows to the brain again.
Which means oxygen and all that. So just a footnote question on that. You say trained, and you say these defibrillation devices are more ubiquitous now, and more people know about artificial respiration. But suppose they're badly trained or not trained. Suppose they try to use one of these devices without good training and they make mistakes. Suppose they try artificial respiration and they do it wrong. Is this dangerous? I mean, is it worth trying if you don't know exactly how to do it?
So when you call 911, EMS, when the operator answers the phone, they ask specific questions about whether you've had training in CPR, whether there are other bystanders who have had CPR training. If no one is available who has had training in CPR, they can actually talk to you on the phone and try to walk you through chest compressions. Over the years, when I first became CPR certified, which would now be about 15 years ago, the algorithms that we were taught were very, very complicated. And the American Heart Association and other organizations have really learned that if you want to get the lay public not afraid to attempt CPR and trained at least to some ability to be able to perform CPR, you really have to make it very, very simple. And so the newest iteration of the guidelines for CPR really focuses on good chest compressions. And actually for the lay public who don't have any medical training now, they don't recommend doing assisted breathing. Because basically the public, because of mouth-to-mouth resuscitation, they were very squeamish about doing that. And so a lot of the public, because they didn't want to do mouth-to-mouth resuscitation, they didn't do chest compressions either. And so now most of the teaching is geared around compressions only, CPR, where
people are more willing to do that.
It's purely chest compressions, right, rather than mouth-to-mouth resuscitation.
Yeah, it's a stranger, they don't know what he's got in his mouth.
Right,
and so
exactly,
forth.
exactly. And so with that very simplified training, CPR can be quite easy to do for the lay public. And it's better to get untrained CPR rather than no CPR.
Okay, okay, so it's worth doing. It's worth being a good Samaritan.
Absolutely.
You know, you wonder about the law in this. If you make a mistake, I mean, I remember there were cases, probably bad law, but there were cases where somebody tried, didn't do it well, and then got sued. What an awful result that is, you know, to discourage people from saving lives.
Right, absolutely. So Hawaii, most states have what are called Good Samaritan laws, and Hawaii is one of those states, which basically means that if you're making a good faith effort to do the best that you can to help somebody who's in medical distress, then you're protected against lawsuits.
Yeah.
That pertains to medical professionals as well, not just the lay people.
Good. We want that. We want that. You know, one thing that I don't think people know exactly how frequent in our society, in our community, there are incidences of stroke and heart attack. I mean, it's the single most important, most prevalent killer, isn't it? Heart attack, And so, you know, it's likely in your life, it's a fair likelihood that you're going to run into somebody who is having a heart attack, and you could help. So it really pays to, A, know how to do that, and B, if, you know, having a defibrillator in your business or your office or whatever it is. Don't you think?
Yeah, that's absolutely a great point. AEDs, or automatic electrical defibrillators, have become very cheap. You can buy an AED for as cheap as $300 off the shelf at this point, and that's a very high quality device. And so you make a very good point among business owners that to protect the public and your customers who are coming into your place of business, or your employees in your place of business, it's worth investing in one of these products.
Okay, we can take
I don't get any kickbacks from AED either, so.
Who are the big manufacturers?
Zoll is a big manufacturer, and there's probably a dozen manufacturers at this point.
That's great. Thank you for helping me with that. That's Matthew Koenig, MD, at Queens Hospital and the John A. Burns School of Medicine. He's doing MRI research here in Hawaii, the state of science. We'll be right back after this short break.
Aloha, I'm Hunter Hevlin, host of Sustainable Hawaii here at ThinkTech Hawaii. You can tune in every week on Thursday at 2 p.m. to see interviews with sustainability professionals from around the state and even further abroad, learning about activities with water management, food security, waste management, and a whole host of other fascinating opportunities to get engaged with making a greener island. So if you're interested in making the transition from consuming individuals to communities of producers, check us out every Thursday.
Okay, we're back. We're live. We're here with Dr. Matt Koenig, and we're talking about Hawaii, the state of science, medical science, and research. We're talking about MRI research at Queens Hospital in conjunction with the medical school of JAPSOM. One thing I want to just also ask you about is when you having the stroke, for example, or a stoppage in your heart beating for any reason, you're depriving your brain of oxygen, of blood, and therefore oxygen. And then you have, you know, an immediate deterioration, and at some point it becomes permanent. Am I right? How does that work?
That's right. So when when blood flow stops to the brain, a person immediately loses consciousness. And brain injury, permanent, irreversible brain injury, begins to set in within minutes of stoppage of the heart. Within 4 to 5 minutes, permanent brain injury begins. And that occurs really because the brain has no mechanism of storing energy. It has no oxygen stores, it has no glucose or sugar stores. Mm-hmm. And so you really need a constant supply of oxygen and blood to the brain in order for it to function. And so even very short minutes count when a patient has a cardiac arrest. That's why it's very important to have bystander CPR and availability of defibrillators in the community.
Yeah, people don't realize how important the brain is in their body as an organ. What did I hear? That the brain requires 600 calories all by itself every day.
That's right.
You said that, I think.
I don't know if I said that, but
okay,
that's true.
Somebody recently said that. And then, of course, you really need to have the oxygen all the time. And now, what about the process of the deterioration? In other words, the process that sets in after a couple of minutes. So let's assume no oxygen, no blood, no oxygen, and now these brain cells, how many billions of brain cells, are beginning to die off. And they probably die in a certain sequence according to how close they are or how much oxygen they need on an ordinary basis. How does that work? Where does it die off first?
So the neurons or brain cells exhibit a quality that we call selective vulnerability, which means that specific neurons require much higher blood flow and much higher oxygen supply, and they are selectively vulnerable to ischemia. Ischemia meaning lack of blood flow. And so the most famous region of the brain that's injured when blood flow is stopped is in the hippocampus. And that's an area of the brain that serves a function in memory, in encoding memory. And so in patients who survive cardiac arrest, those with the least amount of injury, let's say they don't make if a person does not make a complete recovery after a cardiac arrest, which is common.
In terms of his brain.
Right. If the brain does not make a complete recovery, the most common pattern of injury that I would see is memory dysfunction, inability to encode or create new memories.
So, Oh, so, whoa, no, this is more, it's not just that you can't remember things. It's that your hard drive is messed up. You can't put new memories in the hard drive.
That's right.
That's serious. That's more, I mean, you can always, somebody can tell you what happened, so now you can have a new memory of it, but here, you can't have a new memory either.
That's correct. So it's not, it doesn't create a retrograde amnesia where you lose old memories. And the working memory, meaning like if somebody tells you a telephone number and you rehearse it to yourself in your brain in order to remember it before you dial it, that's working memory. The working memory is intact, but the ability to take that immediate working memory and then encode it in the brain so you could retrieve it later, that's what's lost.
It's kind of cruel, I think. You go through your whole life, you get older, you become more, subject to the possibility of this calamity, and your whole life is really a sequence of memories, some of which, as you get older, you can remember easier from way back when, some have not. But I mean, it's kind of interesting that your life is kind of an accumulation of all those memories. Now you don't have that anymore. It's like your scrapbook is wiped out.
Yeah, it's a very, it's really a devastating injury to the brain. And so as a neurointensivist, as a neurologist who takes care of critically ill patients, I'm very often called upon to examine patients who have survived cardiac arrest, meaning the heart has stopped and they received CPR and they were resuscitated and the heart started again and the patient survived to be admitted to the hospital. And what happens in that situation is that 80% of people, or greater than 80% of people who are resuscitated after a cardiac arrest are comatose. And the data is quite poor, actually, for the outcomes from those patients. Fewer than 10% of those patients who are resuscitated are discharged from the hospital neurologically intact, meaning back to normal. And so 90% of people who are resuscitated after cardiac arrest have some type of brain injury. And what's not clear to the physicians who treat those patients or their families, or to me when I see those patients, is here's a patient who had a cardiac arrest who was successfully resuscitated, but now is in coma. Will that person be one of the 10% who will make a good recovery and get back to normal or regain most of their function?
Not likely.
Or will that person have some form of brain injury? And if so, how severe will that brain injury be?
So the whole thing is like a time frame. It's like a stopwatch kind of thing. So the fellow falls down from a heart attack. It's a real deal. It's not one of those slow jobs, right? I mean, we're talking about the kind that happens, right now. And you can watch the stopwatch. 2 minutes, 3 minutes go by, 4 minutes. The ambulance isn't here yet. Nobody has done CPR on him. There's no defibrillator in town, in that place. And tick, 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 he's losing some cells.
That's right.
And the longer that goes on, when does it get really serious? When is, like at 10 minutes, is he a goner?
It really depends on it depends on a number of factors. It depends on whether CPR was started in the field and whether that person was trained or untrained. It depends on whether the arrest was witnessed or unwitnessed, meaning if the person was found just on the ground unconscious and it's not clear when the arrest occurred, that portends a poor prognosis. And so those are the major factors. And also the other big factor is what the initial rhythm was. Was it just that the heart was flatline, what we call asystole or absence of electrical activity? Or was there a pattern called ventricular fibrillation or ventricular tachycardia, which is what we call the shockable rhythm?
Where there is some blood flowing, it's just not in the right rhythm?
That rhythm, meaning shockable, meaning that automatic defibrillators will shock that rhythm. And in many cases, that shock will convert the patient back into a normal perfusing rhythm, meaning a heart rhythm that is able to sustain blood flow to the brain.
But one thing that strikes me, another footnote question, if you don't mind. So we have this process where the brain cells are dying, and you can stop that process by restoring the blood and therefore the oxygen to the brain. But I'm wondering if a lot of people are walking around right now today for one reason or another, one medical reason or another, they don't have enough flow to the brain in the ordinary life. And they don't have enough oxygen to the brain in the ordinary life. And their brains don't work as well because of that. and whether it's possible to know that and whether it's possible to actually fix it. If I walked around with a tube of oxygen up my nose every day, I'd be smarter, wouldn't I?
Not necessarily. The brain has an interesting relationship with oxygen. Certainly hypoxia, meaning low oxygen to the brain, could cause injury over time. That's very clear. But what also seems to be the case is that hyperoxia, meaning excessive oxygen to the brain, especially in the setting of an underlying injury, brain injury like cardiac arrest, is also injurious. And the reasons for that are complex and have to do with oxygen-free radical species.
Chemistry.
Yeah, and other secondary injuries to the brain.
How interesting.
So I wouldn't recommend buying an oxygen tank just
Okay,
yet.
all right. Okay.
But your point is well taken that chronic, that lower blood flow to the brain chronically over time certainly is associated with abnormal aging and poor cognitive function in older age. And the inverse is also true, that people who, this research comes out of exercise research, that people who exercise, that does not just increase blood flow to your muscles, that also dramatically increases blood flow to your brain. And so people who engage in regular cardiorespiratory exercise have much higher blood flow to the brain, and there's a lot of science showing that those people live longer and are more cognitively intact and have a lower incidence of dementia.
So you can control all of that by what you do.
Right.
So I want to get into the whole question of exactly how you conduct your research on exactly what issues in this general area. So let me make a guess and say that at some point you want to examine this person's brain, just like Linda Chang examines it for HIV, and you want to see some before, after, and maybe a lot of snapshots in the middle to see how the brain is changing because of some effect in the body, maybe a stroke or a heart attack. What is your research, doctor, and how do you do it?
So the research really comes out of the clinical question of here's a person that I'm seeing, that I'm consulted on, who has survived a cardiac arrest but is in coma. And the physician taking care of that patient and the family are asking the question, is this person going to recover? And if so, how will this person recover? Or what injury will be present if this person is to survive this injury? And in that situation, the brain is really a black box because the patient is in coma and they may have some reflexes that I can examine at the bedside, but you can't see the brain. You can't really test the function of the brain at the bedside. And so the research really stems out of that very frustrating and frequent clinical situation of seeing a patient in coma and being unable to determine whether this is somebody who can recover or not. And so basically what functional MRI does, let me maybe take a step back and talk about what MRI is, okay? So MRI is
Magnetic resonance
imaging.
imaging.
Right, so magnetic resonance imaging. So basically what MRI does is it uses a very powerful magnet to examine the movement of water within structures, including the brain. MRI can be used to look at other organs as well, but for my research, it's primarily used to study the brain. And basically what you're doing is you're imaging water molecules within the brain, within different structures. And so the appearance of those water molecules is different depending on what tissues they're in. And so MRI primarily, since its inception and its common clinical use, is not used to study brain function. It's used to study brain structure. It's an excellent way of imaging the brain, of seeing pictures of the various structures of the brain and to see whether injury is present. And so in a stroke, which is a permanent blockage of blood flow to part of the brain, it's very easy to see the damage to the brain because there's a wedge of tissue that received blood flow from that artery that's now blocked, and that wedge of tissue is injured. And so
you can see that in the water molecules because they look different.
Absolutely, absolutely. So that picture of the brain very clearly shows an injury. Cardiac arrest is different, though. Cardiac arrest is not a stroke. Okay, a stroke is where you have permanent blockage of blood flow to part of the brain, and that chunk of tissue then dies. Okay, cardiac arrest is a model of injury that we call no flow reflow. No flow meaning there's a period of time where there's no blood flow to the entire brain, not just a segment of the brain, but the entire brain. And then because of resuscitation, that blood flow is then restored after some minutes. So it's a period of no flow and then a period of reflow.
So when
of
you
blood
say
to the brain.
functional, you mean it's more than a snapshot.
Right.
You're
That's right.
checking it at different moments in time, depending on what's happening to that patient. Wow, is this interesting? Aren't you glad you're watching this show? You're learning stuff that nobody knows. This is great stuff. That's Matthew Koenig, MD, at Queens Hospital and at the John A. Burns School of Medicine here in Hawaii, the state of science. We're talking about MRI research, and we're learning so much. We'll be right back after this break.
Hi, I'm your host on ThinkTech Asia, Bill Sharp. I look forward to you joining us each Monday between 4 and 5 o'clock when we film right here in our studio in downtown Honolulu. The show, ThinkTech Asia, focuses on contemporary events in Asia. And by Asia, we mean anything from Hawaii, south to Australia and New Zealand, west to Pakistan, and as far north as the Russian Far East. Clearly, this is one of the most economically dynamic centers of the world, and we bring you up to date on what's going on in a whole host of countries in this very vital region. We look forward to seeing you. Aloha.
Okay, we're back. We're live with Matthew Koenig, MD, at Queens Hospital and the John A. Burns School of Medicine here in Hawaii, the state of science. And we're talking about MRI research. And our engineer has asked me to ask you where weight falls in all of this.
Well, certainly weight, diet, and exercise, which are all interrelated, are risk factors for any type of vascular disease, meaning hardening of the arteries. And that places one at a higher risk for heart attack, stroke, peripheral vascular disease, kidney disease, and cardiac arrest. So clearly weight is a risk factor for all of these conditions.
Mm-hmm. That's happy news. Okay, so I want to get back to the science. So here we have identified what functional MRI research is, and we know it's a series of snapshots depending on certain functions. And so then we want to know from you, doctor, exactly what your research is, and how you use the functional MRI to learn things about the way, the brain works in being deprived of oxygen because of stroke or heart attack.
So the problem with using standard MRI in cardiac arrest, meaning structural MRI, is that you don't see the injury that occurs in the human brain related to cardiac arrest because of this condition we were talking about just before the break, that there's a period of time of no blood flow and then blood flow is restored. And what happens is that the neurons, meaning the brain cells, not the support scaffolding of the brain, but the actual brain cells, the neurons, are selectively vulnerable to that injury. And so what happens is the neurons themselves die, but the rest of the architecture of the brain is preserved. And so in a standard structural image, it has very good resolution, but you really can't see on the microscopic cellular level that the neurons have died. And so
How big are they? They're microscopic.
They're
really
microscopic,
tiny.
yeah, they're microns.
It's the smallest kind of cell you could find in the body.
Mm-hmm. Well, they're
smallest.
They're actually neurons of various sizes, but
Okay.
suffice it to say that a standard structural MRI is not going to pick up that kind of resolution, that you would actually need to look at it. You would need to take a piece of the brain and put it on a microscope and look at it to see the injury that occurs after cardiac arrest.
This is hard to do with a living person.
That's very hard to do, and I don't recommend it. And so, as a clinician who treats patients who have survived cardiac arrest and attempts to predict who will wake up and not wake up, it's been very frustrating because the structural MRI can look totally normal in a patient who has a devastating brain injury. And so using functional MRI to study this condition is an attempt to take that very useful clinical test, MRI, and view it in a different way to gain more information, not just about the structure of the brain, but about how the brain functions, both during the injury period after cardiac arrest and the recovery period.
So you have to see how the normal path works and then see how different aberrant paths work and see what path is being taken at a given moment in this patient.
That's right. And functional MRI really, it takes advantage of a phenomenon in the brain that couples blood flow to function. And so parts of the brain that have a higher level of function at any given moment also receive higher blood flow. which sort of makes sense, right? If a part of the brain is more active, it's kind of like exercising and it's demanding more blood flow.
Calling for it. Yeah.
It's like if you're, doing bicep curls, you're going to have more blood flow to your bicep while you're doing that. The brain is doing the same thing.
Isn't the body amazing that a cell or a bunch of cells can command more blood and have it delivered?
Absolutely. And the really amazing thing is how quickly that happens and how quickly that changes. I mean, within tenths of seconds, the blood flow to the brain is changing related to how it's functioning. And so the simplest example of functional MRI is if you had a patient in the scanner and you had them move their right arm. And you took a snapshot of the blood flow to the brain while they were moving the right arm. And then you had them stop moving the right arm, and you took another snapshot when they were not moving the right arm. And then you did that repetitively, and you summed together all the images of blood flow to the brain during that movement, and then subtracted out the blood flow to the brain when the patient was not doing that movement. What you would be left with is just the blood flow to the brain that occurred with the movement. And that's exactly what functional MRI does, is it highlights the part of the brain that's receiving the most blood flow during that movement. And so in that example, it would point to the motor cortex, or the part of the brain that controls movement.
So in that case, it's motor then. So what you're looking at is the part, there's so many things that flow out of this, no pun intended. So if it's motor, then the motor neurons, which are calling for the movement of that arm, are commanding more than the baseline. And so you can see how much they are commanding and how much is being delivered to them is really what you're looking at.
That's right.
In the brain because of the super MRI. Okay, so now I know what this extra overlay of command of oxygen is to those special neurons. What good is that, in terms of the result of your research?
Right, so how do you apply functional MRI, which I just described in a very simple example of a patient who could respond to a task, like move your right arm? How do you apply those principles to a patient who is comatose? And by comatose, I mean completely unresponsive to the outside world.
And that's flatline.
Right, not necessarily flatline per se,
when I mean flatline, I mean the baseline of blood to the brain.
Right.
Without movement.
Right, for all intents and purposes, if a person is in coma or appears to be in coma, meaning is not consciously responding to the outside world, then clearly the example that I just gave of asking them to move their right arm to command, right, would not be a good test for that person. And so really, how can you use functional MRI to study an unresponsive person? Well, fortunately, a lot smarter people than me over the years have developed a technique that's called resting state fMRI. That's one of the most common names for it. And basically, resting state fMRI is the changes in MRI that occur at rest. Meaning the example I gave of asking the patient to move their arm and then not move their arm. And as I said, you would just subtract out that baseline signal of the brain to look at the part of the brain that is most active during the movement, right? So rather than taking that no movement data and just throwing it out completely, there's actually a lot of information in there. Because what's in there is the brain at idle, when the brain is just idling with no specific task. And I've actually been a subject in functional MRI studies. I was, as a starving medical student, I picked up some pocket change doing fMRI research.
What's the F stand for?
Functional, yeah,
Okay.
fMRI, functional MRI. And so I've actually been a guinea pig in my own, I actually did some for my own project, and I've done some over the years. And so basically I know that the researcher asks you during the fMRI task, just lay in the scanner and just empty your mind, right? But that's of course impossible to do, right? You don't lay there in the fMRI scanner and think of nothing, right? You lay there and think of, what am I gonna do later today? I'm hungry, I recount a conversation I had earlier in the day and think about how might have gone differently. And we might call that introspection, right? That's your sort of idling thoughts of the brain.
It's something, it's something above the true baseline.
Right. It's certainly not an empty mind. And even if you're told to lay there and think of nothing, it's really impossible to lay there and think of nothing.
Yeah.
Right? There's really a process of introspection going on, which involves memories and social planning and recounting things to oneself.
Well, if you get really, really fine-tuned about this, you can say, well, if I'm idling about a trip to France, that's one level of activity. If I'm idling about having lunch, that's another level. And maybe I can distinguish this is true? Distinguish
Oh, absolutely. Absolutely. I mean, you can actually do tasks that are not outward tasks. And a famous example is a research group in Cambridge who does a lot of very high-level functional MRI research in patients with disorders of consciousness like persistent vegetative state. They do a task where they ask seemingly unconscious patients, or patients who are not interacting with the outside world, they ask them to imagine themselves playing tennis. And a subset of those patients have activity that occurs in the motor planning area of the brain, where one might imagine themselves playing tennis. Another task is that they imagine patients to imagine themselves walking around their house. They ask the patient to imagine themselves, walking around their house, and then the part of the brain that's engaged in visuospatial relationships lights up in a subset of those patients who otherwise seemingly appear to be unconscious.
Can I read thoughts this way? I mean, not today, not right now, but ultimately, will I be able to read thoughts?
I think so. I mean, I think fMRI research is not really ready for that kind of single subject, real-time answer, because there's still a lot of processing that needs to be done. And I mean, even in my research study, which does not ask patients to do these specific tasks, it's actually just a resting state, meaning the patient is not asked to do anything. But even in that research, the only way to really get at the signal is to collect a number of patients, say 10 patients or a dozen patients, compile that information together, and then subsegment it to look at the individual patients. And so the ability to look in real time and read someone's thoughts with the fMRI scanner on an individual patient in real time is not quite there yet. Mm-hmm. But it's fairly easy to see how, as this research continues to progress, that it could be used in that way. And there are groups that are claiming, to use it as a lie detector test or use it to study people's feelings towards things. And so that research is certainly evolving.
When you talk about MRI, magnetic resonance imaging and all that, I've heard about invasive objects, sensors, for example, tiny sensors that are, planted in the brain, mm-hmm. that can either read what's going on or can make what's going, can actually create thoughts or motor activity. Where's this science going to go? You think it'll hang around, continue to operate with MRI, or will we have different kinds of sensors that will tell us more?
Yeah, that's a great question, and it's actually very relevant to my own research past. Before I moved to Hawaii and began doing fMRI-based research, I was actually a translational researcher using an animal model of cardiac arrest. And in that situation, we weren't using MRI to study brain recovery. We were actually using implantable sensors, as you mentioned. And those are called multi-unit probes. And basically, you place probes into the brain of the animal. In our case, we were using rats. So you place probes into the brain of the animal, and it actually detects directly the function of neurons by what are called local field potentials.
Different way of sensing.
A different way of sensing, exactly. More of a direct measurement of electrical activity of neurons, whereas fMRI is kind of one step removed from that. It's non-invasive, which obviously is why you'd want to use it in humans, but it's not directly studying the activity of neurons. It's indirectly studying it by studying blood flow to those regions.
So here's my follow-up question, or my last question for you. So you got all this neurology training. You've got all this research under your belt. You're working in what I consider a fine laboratory with great mentor and all that, using great equipment. But as time goes by, you may find the technology goes in the other direction. You may find that despite all your time and effort and writing, I presume, about the subject of functional MRI, you may find that there are other sensors that can sense more and give you a better picture of exactly the same kinds of things you're looking for in dealing with health. How hard is it for you? How hard would it be for you to get off the one research track and onto the other?
Well, that's kind of the exciting challenge about doing research, really. And that's sort of what I did. I went from an animal model of cardiac arrest with invasive sensors. That really, if you take a step back from it, all I was studying was network activity of the brain, how different parts of the brain communicate with one another using an animal model and using a sensor. And then after relocating to Hawaii and working with Linda and Tom in their lab, translating that into humans, not an animal model, but actual clinical care of patients, and using functional MRI, which is a totally different means of studying the brain. But in actuality, I'm really just studying the same thing. I'm studying network activity of the brain. I'm studying how different regions of the brain communicate with one another in a network. And so that's the exciting thing about science, I think. There's serendipity and there's advancement in the field, and you just have to be open-minded and have the right mentorship and support. And it is surprisingly easy to translate those basic concepts into different methods of study.
That's great to hear. That's great to hear. Actually, I feel my brain has improved during our discussion.
Me too.
Yeah.
My brain.
That's Dr. Matthew Koenig of Queens Hospital and John A. Burns School of Medicine here in Hawaii, the state of science, doing MRI research at Queens and Japson. Thank you so much, Matt.
Thank you.
Aloha.
Thanks a lot, Jay.